The series records in the GEO database for RNA-Seq often include a text file containing gene counts in the supplementary file section. In such cases, it is not necessary to process FASTQ files yourself. If a supplementary gene count file is unavailable, you can still generally obtain gene counts from GEO. Visit our website to find the URL. Go to Help and search GEO FASTQ. Click Getting FASTag GZ files from GEO. Copy the URL provided in the instructions. Modify the series ID to match your dataset, then access the file in your browser. The gene counts matrix generated by NCBI is available in a fixed format, calculated using a standardized protocol. On the other hand, the supplementary files format and protocol vary because they are determined by the authors. Now we have two gene count tables. Uncompress the GZ files. Delete the GZ files after uncompressing. Open the GEO-generated version table in Excel. The gene IDs are NCBI, NTRES, gene IDs, and the sample titles are GEO sample accession numbers. The supplementary files gene IDs are ensemble human gene IDs, and the titles are the author's naming. We first import the GEO-generated version data into the Subio platform, because it's standardized. Before importing samples, we create a platform of NCBI human gene IDs. Get the human gene info file from the NCBI FTP site. Uncompress the GZ file, and open the gene info file with Excel. Select the whole table, and sort it by the type of gene column. Delete all biological region rows. Delete from unknown to tRNA rows. And delete rRNA rows as well. Overwrite the file. Open the Subio Platform's Data Manager tab. Click New Platform. Drag and drop the edited gene info file to the panel. Select the gene ID column for the measurement ID of this platform. Next, click Import Samples, then drag and drop the GEO-generated gene count table. Extract individual samples by clicking Multiple Samples in one file. Complete the sample import wizard. The imported samples are labeled only with GEO accession numbers, which are hard for humans to understand. So get the soft formatted family file to annotate samples. You don't need to uncompress this file. You can take this file from the lookup tool of the Edit Sample Information panel. The file name and GEO accession do not match directly. To align them, split the file name column at the dot to remove txt, ensuring the remaining part corresponds to the accession. Open the lookup tool again. Drag and drop the soft file to the tool panel. Select key columns to match. Select all columns to import, and finish. Delete the temporary column and save. Hide all columns and select columns to display. Now it's ready for analysis. Create a series of the imported samples. Next, we import the supplementary file version of gene counts to the Subio platform. Remember that this table has different gene IDs and sample names. A platform for ensemble human gene IDs is ready as a preset platform. Take the supplementary file version of gene counts to the Import Samples wizard, and extract samples using the Multiple Samples in One File button. Complete the wizard. Samples are labeled with the author's name. Remove .txt to extract the genuine names. The sample titles of GSM records have endometrial biopsy before the names. So, edit the names with Excel to fit the sample titles. Remove the temporary column, then open the lookup tool. Drag and drop the soft file to the lookup tool. Now both tables have matching key columns. Select all columns to import and finish. Hide all columns and select only the columns to display. Now, create the series of supplementary gene counts.